Hi, Doctor. My name is Trent. I'm the group leader for our research topic, which is visual attention patterns and eye tracking analysis in Gen AI images. So let's move on to the introduction. So basically, the issue or the problem statement we're trying to address is basically Gen AI images outputs are highly realistic nowadays. This blurs out visual distinctiveness from authentic images. So basically, even the most existing studies due to that is basically mainly focused on image realism and AI detection accuracy. Because of that, there's lesser attention given to gaze behavior, visual attention patterns, stimulus difficulties, and gender differences using image comparison task. So our research basically we asked these four main research questions, which is number one, which image pairs receive the greatest visual attention during comparison. Number two, is participant correctness associated with different eye tracking patterns? Number three, do difficult image pairs yield divergent visual attention matrices compared to easy pairs? And the final research question is basically, do male and female participants present distinct eye gaze patterns? So before moving on to our measurable objectives of the research, I want to go through the hypothesis that we proposed according to the or based on the research questions. The first hypothesis is basically specific image pairs yield higher processing matrices. Second, gaze structure differs between correct and incorrect responses. So participants who got the image detection correct, their behavior is usually different compared to those who got it wrong. The third hypothesis, longer fixation duration in difficult image due to high cognitive process demanding. And the final hypothesis, difference in gender results in difference in visual attention patterns during tasks. So the measurable objective for our research is basically the first one, quantify task attention. So we check mean fixation duration and total fixation counts across 6 visual stimuli. Second one, map cognitive demands. So contrast descriptive case benchmark across varied 6 visual stimuli that we use. And the final one is model participant subcohorts. So we evaluated variation factors between correctness categorization and also our demographic. So the main demographic, obviously, we all students. So the main demographic that was used is basically we differentiated between two gender classes.
Moving to our literature review, we analyzed 4 key studies that directly inform our research question. First, Huang and colleagues 2024 work on style gantry eye tracking validation paradigm found 76.8% accuracy and noted closer scrutiny and suspected AI elements. While limited to synthetic facial features, these studies supports our first research questions regarding how stimulus features draw user gaze. Next, Kotovich's 2010 study on gaze contingent search trial demonstrated the fixations index confidence. Though their targets were abstracted rather than real scenes, these guides our second research questions evaluating correctness versus duration profiles. Third, Edridge-Squad's 2016 research on cognitive load established that average fixation duration is a reliable index of difficulty. This validates our third research question by justifying duration as a cognitive load indicator. Finally, Vassallo and colleagues in 2009 assessed observer sex differences and found elevated scan path durations in male participants. Although restricted to facial affect recognition, this provides the empirical basis for testing gender differences in our fourth research question. Let's look at methodology and architecture. For our participants, we recruited a total sample of 129 university students, volunteers under control parameters. The group consists of 87 male participants making up 67.4% of the sample and 42 female participants making up the remaining 32.6%. In terms of memory and hardware, we utilize a Adobe Pro Tracker unit configured to screens with a 1920 by 1080 resolution. We recorded the participants' gaze points using standardized 5-point calibration method. Our procedure followed a structured step-by-step flow. We started with our orientation briefing, followed by a calibration checklist and practice trials. The core experiment involved 6 experimental image pair screens labeled T1 through T6 that were displayed side by side for up to 15 seconds each.
All right, for the pre-processing, basically we go through a few steps, which is data ingestion, trial filtering, relational merge, ID normalization, cleaning cohorts, AOI partitioning, and also metric aggregation. And we extracted a few feature vectors, which is mean fixation duration, total fixation count, AOI level spatial split, gender group mean, and correctness group. For the software that we use Tobii ProLab trial version, Python Pandas, Python Matplotlib, Python OpenPyXL, VS Code, Git, GitHub, Google Sheets, and finally the analysis methods are basically descriptive statistics, AOI spatial orientation, correctness link gaze, difficulty comparisons, and also gender gaze comparisons.
Alright, let's get into the results. I'll start with average fixation duration, which is basically how long participants held their gaze on each image pair before moving on. The standout here is T4, the pen image pair, which came in at 384 milliseconds, the highest of all 6 stimuli. Right behind it is T5, the fish pair, at around 329 milliseconds. The rest, umbrella, ring, prisoner, and heart, all clustered between 268 and 299 milliseconds. So why does this matter? A longer fixation duration generally signals that the brain is working harder to process what it's seeing. T4 and T5 clearly demanded more of our participants' attention. Now, if we flip the total fixation count, which is how many times participants look at each stimulus, we actually get a very different picture. T4, the heart pair, had the highest number of fixation at over 212,000 But here's what interesting. T6 also had the shortest average duration, and T4 pen, which had the longest duration, had the lowest count at around 140,000. So we are seeing a kind of inverse relationship. T4 prompted fewer but much longer looks, which means a deep and concentrated scrutiny. T6 triggered a lot of quick glances, like a broad, rapid scanning. It's two different cognitive strategies. We also look at whether participants paid more attention to the left or right of the image in each pair. The left side got more fixation overall, about 1.9 million versus 1.77 million on the right. But the right side had a longer average duration, which is about 608 milliseconds compared to 550 on the left. So participants were jumping to the left image more often, but when their eyes landed on right image, they stayed there longer. Next, we group stimuli by difficulty. Easy pair, T1, T3, T1 prisoner, T3 umbrella, T6 hard, average around 280 milliseconds on fixation. Difficult pairs like T2 rings and T5 fish, average about 312 milliseconds. That supports the idea that harder stimuli really do increase cognitive load. For gender, male participants average about 313 milliseconds, females average 300 milliseconds. Descriptively, males held their gaze slightly longer, but we didn't run any inferential tests like a t-test, so we can't make a strong statistical claim from this. Wrap up the results. Our key takeaways are that T4 pen was the most cognitively demanding stimulus based on fixation duration, while T6 hard triggered the most frequent gaze activity. Harder image pairs consistently produce longer fixations, and we found an interesting left and right asymmetry in how participants scan the screen. One thing we weren't able to fully resolve is the link between correctness and gaze patterns. Accuracy data was collected, but it wasn't statistically merged with the eye tracking metrics. That is something we think is a clear direction for future work, maybe through a logistic regression.
Okay, next we have discussion, cognitive explanation. Fixation inverse relationship, duration versus count. T4 pen exhibit the longest mean duration, but the lowest count. Meanwhile, T6 hard showed the opposite profile. This suggested that T4 required deep, highly concentrated scrutiny, fewer but longer looks, whereas T6 prompted broad, rapid visual scanning, higher frequency, brief looks. Also, the attention and difficulty metrics. Visual region asymmetry. Gaze point shifted frequently towards the left screen region, but participants rested their eyes on the right side images for longer duration. Stimulus difficulty. Difficult image pairs, T2, T5, averaged 311.98 milliseconds per look, whereas easy pairs averaged 280.03 milliseconds. Elevated complexity correlates directly with cognitive processing time. Also, limitations and future directions. Project limitations such as analytical scope, lack of spatial AOIs, visual outputs, and also recommended pathways, implement differential tests, map micro AOIs, and also demographic expansion.
So Taufik. So to conclude, the study successfully applied eye tracking techniques to investigate visual attention patterns where participants compared AI-generated and human-generated images. Our findings show that different stimuli attracted different level of visual attention. We observed that difficult image pair generally resulted in longer fixation duration, suggesting greater cognitive effort during visual processing. We also found that fixation duration and fixation count capture different aspects of attention, providing valuable insight into how participants interacted with the visual stimuli. Overall, the results demonstrate that the eye tracking is an effective tool for studying visual perception and attention in Gen AI related tasks. For future work, the study can be expanded through the uses of AOI, heat maps, campus visualization, and statistical testing to provide deeper insight into user behavior. We would also like to express our sincere appreciation to the course coordinators of WID 2003, Cognitive Science at the Department of Artificial Intelligence, University of Malaya, for facilitating access to the Tobii hardware and software suite. We thank all peer respondents who volunteered their session to populate our tracking database. This slide shows summarize our contribution to the team. Throughout the semester, our team completed key stages, including topic selection, literature review, data collection using Tobii, eye tracker, data analysis, report writing, presentation preparation. The Gantt chart illustrates how these tasks were scheduled and coordinated to ensure the project completed on time. Finally, this slide summarizes the contribution of each team member. Each team member was assigned specific responsibility, including research and planning, presentation slides, report writing, report and coordination, video editing, and etc. That's all from us. Thank you.